You think you know what glows in the ocean, fireflies. Maybe glow sticks at a rave. But down there, where sunlight dies at 200 meters, 3/4 of everything that moves makes its own light. Not reflection, not tricks. Chemical fire inside living flesh. And these 7 creatures do it better than anything else you will see in your lifetime. Scientists at Mbari spent 17 years filming 390,000 individual observations off the California coast, from the surface down to 3,900 meters. Published April 4th, 2017. In scientific reports, they confirmed 76% of observed organisms possess bioluminescence capability. That is not a typo. 76%. The ocean is a light show, and most of it happens in total darkness. The mesopelagic zone, between 200 and 1,000 meters, hosts some of the dimmest habitats in the world. Yet down there, ambient light is enhanced by a multitude of bioluminescent signals, dim, intermittent, but everywhere. Brittle stars, squids, fish. The chemical reaction involves luciferin molecules oxidized by an enzyme called luciferase in the presence of oxygen. That is the universal mechanism, but each creature tweaks it, modulates it, weaponizes it differently. Now we are going to walk through the 7 most spectacular performers. No fluff, just names, depths, mechanisms, and the numbers that matter. If you value documented marine biology and deep sea history like this, subscribe. It is the best way to support our work. Also comment from which city you are hearing this from. This script is researched and factual, using sources from a March 24th, 2024 comprehensive species level inventory, published in the journal Life by researchers Du Châtelet, Deloise, Flamand, Mahillon, and Malafeit. That paper cataloged marine luminescent animals and showed the world's luminescent organisms now encompass around 1,000 genera, a significant increase from previous estimates. We also drew from May 10th, 2024, research published in Biological Reviews analyzing bioluminescence in deep-sea decapod shrimps, and from genome sequencing published in Scientific Reports in 2016, identifying crystal proteins in firefly squid. Photophores. Now back to the information. Commercial catch from 1990 to 1999 ranged from 4,804 to 6,822 tons annually. That is tonnage, not specimen count. Blue and green bioluminescence from the body photophores, only blue from the eye and arm photophores. The blue light peaks around 470 nanometers. The green light appears in 10 to 18% of body photophores, with 82 to 90% producing blue. The crystals contain 3 proteins, bioluminescent, 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 all members of the ANL superfamily of adenylating enzymes. They share 19 to 21% sequence identity with firefly luciferases, but use a completely different substrate, cholinterrazine disulfate. The squid is the only organism known to produce light using protein crystals. The luciferin and luciferase sit in crystalline structures within rod-like bodies in the photophores. These microcrystals measure 0.6 to 2 micrometers. The optimum temperature for the luminescence reaction is about 5 degrees Celsius. Cholinterrazine disulfate is a strictly specific substrate. Any modification of its structure results in very heavy loss in light emission capability. This directed cone of bioluminescence allows the squid to detect prey and predators from below and attract small fish to eat. The 3 large photophores at the arm tips flash with the brightest light, as if the squid is waving burning flares. 5 smaller photophores surround each eye. Hundreds more dot the length of the body. The arrangement creates complex patterns used to disorient predators and attract prey. Research from 2008, published in Biochimica at Biophysica Acta, investigated the ATP-dependent luminescence reaction catalyzed by membrane-bound luciferase in detail. Mated females stored an equivalent amount of sperm in both pouches surrounding their seminal receptacles. They observed a gradual decrease in sperm quantity during the reproductive season, indicating preservation of sperm through the lifespan of the female. A single male sperm fertilized all of the female's eggs. Both findings support monogamous reproduction. Eggs hatch in 6 to 14 days depending on water temperature, which varies from 6 to 16 degrees Celsius. Higher temperatures encourage quicker hatching. At 15 degrees Celsius, 1 hour after fertilization, polar bodies appear. By 5 hours, first cleavage occurs. By 10 hours, 100 or more cells have formed. Around 16 hours, the embryonic lobe has developed. The larvae drift through the water, developing their cloak and ability to glow as they mature into adult squid. Species. This gives them an advantage in the deep sea, where distinguishing between different light sources can mean the difference between finding a mate or becoming prey. The green abdominal photophores exhibited a gradual decay and maintained their glow in the dark. Conversely, when animals were exposed to air, green luminescence alone was quickly extinguished. There were no observed color shifts, such as from green to blue in any photophores. The ocular photophores emit blue light irregularly, partly because they are concealed by epidermal chromatophores. When threatened, they use bioluminescence for camouflage through counterillumination, breaking up their silhouette against faint surface light. The light from photophores matches the color and intensity of downwelling light, rendering the squid almost invisible to predators lurking below. While synchronous illumination supports the Crypsis hypothesis, the ability to independently control each type of photophore could also support a previous hypothesis of conspecific signaling for communication with other firefly squid. The squid are eaten raw, known as hotorika in Japan, or cooked. Raw consumption of infected specimens causes abdominal pain, nausea, vomiting, diarrhea, creeping eruption, and ileus from Spirurina type X larvae, a nematode parasite. But the light display remains one of nature's most intense bioluminescent events. The mature vampire squid also purposefully agitates bioluminescent protists in the water as a luring method. The deep-sea anglerfish, serratioid anglerfishes, suborder Serratioidae, 167 species across 11 families. Most speciose fish suborder in the bathypelagic zone below 1,000 meters. Females range from a few centimeters to over a meter, depending on species. Males are tiny parasitic dwarfs. The female has a luminous lure, the esca, dangling from a modified dorsal ray called the ilicium. The esca contains bioluminescent bacteria, not the fish's own chemistry, borrowed light. Serratioid anglerfishes consist of 167 species from 11 families and are the most speciose fish suborder in the bathypelagic zone. Most female serratioid anglerfishes host extracellular luminous symbiotic bacteria in the esca. The genera Cryptopteras and Serratius harbor bacterial symbionts in additional pouch, like protuberances anterior to the dorsal fin, known as caruncles. Bioluminescent symbiosis is thought to be essential to survival of adult anglerfishes, although the exact function has not been observed directly in the wild. Utilize diverse sugars. They lost most genes for making amino acids and breaking down nutrients other than glucose. The fish supplies nutrients and amino acids. The bacteria retain genes for building a flagellum, useful in water outside the host. Genes involved in chemotaxis and motility that are thought to be useful only outside the host are retained in the genome. Both species of bacteria showed the largest number of transposons ever reported in a bacterial genome, each accounting for close to a third of their genomes. Transposons move around the genome, insert themselves into other functional genes, and make those sequences unfunctional. Part of the process of gene loss, the evolution is happening independently in each lineage of bacteria. Since the bacterial genomes were different, but each showed evidence of evolution taking place, researchers concluded the evolution was happening independently in each lineage of bacteria. Judging by their undeveloped escae, female anglerfish larvae do not appear to have the real estate for luminescent bacteria at a young life stage. Only after a pore develops do bacteria inhabit the lure once it comes in contact with seawater. Female adult anglerfish from the Linephrine family collected in the northern region of the Gulf of Mexico show fully developed escas with active bacterial colonies. Male anglerfishes do not have an escae and therefore are not bioluminescent. Using specialized equipment, the team extracted 183 seawater samples at 4 to 5 different depths, with a maximum of 1,500 meters, at 12 different locations in the northern Gulf of Mexico, with the hope of locating the luminescent bacteria within the approximate home range of anglerfishes. Researchers wonder if fish spew bacteria into the environment once microbe populations grow, possibly ensuring future generations have access in the water. 16 sRDNA sequencing of those 183 seawater samples detected confirmed symbiont sequences at low relative abundance in 13% of seawater samples. Bioluminescent symbiont sequences were absent from larval serratioid samples, but found at all depths in seawater, with highest abundance at mesopelagic depths. As adults spend the majority of their lives in the meso and bathypelagic zones, the trend in symbiont abundance is consistent with their life history. These findings support the hypothesis that bioluminescent symbionts are not present throughout host development and that serratioids acquire their bioluminescent symbionts from the environment. The bacteria use a luciferin-luciferase reaction involving ATP, magnesium 2+, and molecular oxygen. If the fish cannot obtain food and is starving, research from 1976 by Meyer Rochow showed the light becomes increasingly dim until emission stops altogether. The lure attracts prey in dark, deep-sea environments, may also draw in males for reproduction. Females from the Linephrine family collected in the northern Gulf of Mexico possess both a luminous lure and a luminous beard below their jaw. Only about 30 adult female Pacific football fish specimens exist in collections worldwide. One washed ashore in Crystal Cove State Beach, Newport Beach, California. The Natural History Museum of Los Angeles County now holds it. The only other anglerfish from California waters was collected in 1983. 2 more specimens, one from 1975 from Indonesia, from a trawl at 2,700 feet, another from 1971 from Hawaii at 2,100 feet. Green biofluorescence emitted when exposed to blue fluorescent light. The combination of bioluminescence and biofluorescence in deep-sea animals is extremely rare, documented only in one other group of deep-sea fishes, a jellyfish and a siphonophore. The light produced by bioluminescent lure bacteria enables the biofluorescence. Found in all the world's oceans, primary source of flow agitated bioluminescence in coastal regions. Research published March 15, 2024 in the journal Algae estimated bioluminescence intensity in Korean waters for 3 species: Noctiluca scintillans, Polycricos cophoide, and Alexandrium mediterraneum. Noctiluca scintillans caused 125 red tides in Korean coastal waters from 1981 to 2022. Up to 30% of bioluminescence measured at stations in the Sargasso Sea was caused by Pyrocystis noctiluca, according to research from 1985 by Swift. Light emission involves membrane-bound luciferase, catalyzing the oxidation of luciferin, a tetrapyrrole-based molecule. The time from stimulus to light emission is less than 20 milliseconds, one of the most rapid cellular processes known. Each dinoflagellate cell produces more than one flash, lasting on the order of 100 milliseconds. The bioluminescence capacity depletes once all available luciferin has been oxidized. Shear stress exerted on the cell membrane triggers a mechanotransduction pathway to generate an action potential across the vacuole membrane and associated scintillons. Research published September 5, 2013, showed a cascade involving activation of GTP binding protein coupled receptors in the plasma membrane. The action potential travels across the membrane of the scintillon, activating voltage-gated proton channels concentrated on the scintillon membrane. The influx of protons from the acidic vacuole rapidly lowers the pH and activates bioluminescence chemistry. The 2020 red tide is considered the strongest on record since 1900, when scientists started monitoring San Diego plankton. A 2024 study published April 8th in ChemBioChem reported bioluminescence from dinoflagellates at a scale of about 400 kilometers observed from space. Pyridinium bahamense can completely dominate bioluminescence under bloom conditions. Bathy photometers use flow agitation to mechanically stimulate bioluminescence. The Seliger Bathy photometer from 1968 was the first system measuring bioluminescence to quantify abundance of source organisms. It was used in bioluminescent bays of Jamaica and Puerto Rico to measure abundance of Pyridinium bahamense. Laboratory experiments showed dinoflagellate flashes cause a startle response in predators, disrupting feeding behavior and reducing grazing rate. Dinoflagellate bioluminescence also acts as a burglar alarm to attract a secondary predator that threatens to eat the primary predator. When handled by a predator, the dinoflagellate cell flashes due to imparted mechanical stress. Any mechanical stress of sufficient magnitude, such as forces in waves, surge, or swimming animals, triggers luminescence. Recent research published June 11, 2024 in PeerJ described first flash kinetics of Pyridinium bahamense and Alexandrium monolatum, 2 harmful algal bloom species from the Indian River Lagoon, Florida, along with Pyrocystis fusiformis, Pyrocystis noctiluca, and Lingolodinium polyedra. Flash duration, peak intensity, rise time, decay time, and other parameters vary greatly among species, facilitating plankton identification. Most species have 8 strips called comb rows running the length of their bodies, bearing comb like bands of cilia called setaens. When cilia beat, light scatters, producing a rainbow of colors. The beating combs act like a prism, breaking light into color components. This is not bioluminescence, this is light refraction. Bioluminescence is caused by activation of calcium-activated proteins, named photoproteins in cells called photocytes, often confined to meridional canals underlying the 8 comb rows. In the genome of Nimiopsis ladii, 10 genes encode photoproteins, published December 21, 2012 in BMC Biology. These genes are co-expressed with opsin genes in developing photocytes, raising the possibility that light production and light detection work together. Even crystal jellies, where cholinterrazine was first identified, do not make their own, getting it by eating copepods and other crustaceans. Researchers raised several generations of comb jellies in captivity, feeding them only organisms without cholinterrazine. The comb jellies still glowed to produce cholinterrazine independently. This shows how important bioluminescence is for many marine animals. From an ecological perspective, glowing comb jellies provide cholinterrazine to many other deep-sea animals that eat them. The genome of Nemiopsis ladii has been sequenced. This species could be used as a model animal to study bioluminescence and cholinterazine production. Among the earliest metazoans, the group comprising all multicellular animals. They have a maximum length of 5 inches, found in all oceans worldwide. Recently invaded Black, Caspian, and Baltic seas, where they are considered pests because they consume fish larvae. The comb jelly, Nemiopsis ladii, lives off the eastern seaboard from Maine to Florida. Light-emitting photoproteins are located in the same cells as opsin genes, the most primitive type of light detector in animals. All animals use opsins to catch photons of light. Finding a functional opsin in a ctenophore indicates they have been preserved throughout animal evolution. Co-localization may confer an advantage, a feedback mechanism allowing the comb jelly to maintain the right amount of bioluminescence or enhance reproductive or defensive capabilities. They can also reproduce asexually through budding. Few larval comb jellies survive to adulthood as they are eaten by larger organisms. In times of food shortages, comb jellies shrink their body size to conserve energy and pause reproduction, growing and resuming reproduction when food becomes available. Small cephalopod found throughout temperate and tropical oceans in extreme deep sea conditions between 600 to 900 meters. Not a true squid, not an octopus. Belongs to its own ancient order, Vampyromorphida, phylogenetic relict. Only known surviving member. First specimens collected on the Valdivia expedition, originally described as an octopus in 1903 by German teuthologist Carl Chun, later assigned to a new order. Reaches about 30 centimeters in length, dark red to black body. 8 arms connected by webbing resembling a vampire's cape. Soft gelatinous body lacking stiff support. Large reflective eyes, among the largest proportional to body size in the animal kingdom. The eyes can appear red or blue depending on lighting. 2 long retractile filaments located between the first 2 pairs of arms on the dorsal side distinguish it from both octopuses and squids. These filaments have small hairs made of many sensory cells helping detect and secure prey. From undersides of caped arms. 2 larger white areas on top of the head were initially believed to be photophores, but are now identified as photoreceptors. Chromatophores are poorly developed. The animal is incapable of changing skin color in dramatic fashion, as such ability would not be useful at lightless depths. And exposing spiny projections on tentacles called cirri. The underside of the cape is heavily pigmented, concealing most of the body's photophores. Glowing arm tips are clustered together far above the animal's head, diverting attack away from critical areas. This is called the pumpkin or pineapple posture. This luminous barrage may last nearly 10 minutes, presumably serving to dazzle predators and allow the vampire squid to disappear into darkness without swimming far. The glowing ink sticks to the predator, creating the burglar alarm effect, making the predator more visible to secondary predators, similar to the Atolla jellyfish's light display. The display is made only if very agitated due to metabolic cost of mucus regeneration. Bioluminescent fireworks are combined with writhing of glowing arms, erratic movements, and escape trajectories, making it difficult for a predator to identify the squid among multiple sudden targets. Very slow metabolic rate and efficient blood cells, which happen to be blue, not red, allow it to thrive in the parts of the ocean with the lowest concentrations of oxygen. The vampire squid has adapted to waters of the aphotic zone, far below where sunlight can reach. As a sedentary generalist feeder, feeds on detritus, including remains of gelatinous zooplankton such as salps, larvaceans, and medusae jellies, complete crustaceans such as copepods, ostracods, amphipods, and isopods, plus fecal pellets. The filaments have small hairs on them made up of many sensory cells that help detect and secure prey. They combine waste with mucus secreted from the suckers to form balls of food. Vampire squids purposefully agitate bioluminescent protists in water as a luring method to attract larger prey. This is a unique feeding strategy among cephalopods. Its bright red eyes and dark color earned it the mythical name. Those eyes are actually the largest proportionally of any animal, relative to its small size. Blood contains high concentration of hemocyanin, allowing efficient oxygen extraction from low oxygen environments. Photophores are used for counter-illumination camouflage, making it difficult for predators below to spot the squid against faint light from above. Found in oceans worldwide, named after conspicuous use of bioluminescence. Among the most widely distributed, diverse, and populous vertebrates. Total global biomass estimated at 1.8 to 16 gigatons, accounting for up to 65% of all deep sea fish biomass. Commercial fisheries exist off South Africa, in the subantarctic, and in the Gulf of Oman. Range from about 2 to 30 centimeters in length, most under 15 centimeters. Shallow living species are iridescent blue to green or silver. Deeper living species are dark brown to black. Slender compressed body covered in small, silvery deciduous cycloid scales. large, bluntly rounded head, large elliptical to round lateral eyes, large terminal mouth with jaws closely set with rows of small teeth. Single high dorsal fin, forked caudal fin, adipose fin. Anal fin, supported by cartilaginous plate at its base, originates under or slightly behind rear part of dorsal fin. Arrangements of photophores are different for each species, so bioluminescence is thought to play a role in communication, specifically in shoaling and courtship behavior. Concentration of photophores on flanks indicates use as camouflage. In counter-illumination strategy, lanternfish regulate brightness of bluish light emitted by photophores to match ambient light level above, effectively masking silhouette when viewed from below. Well known for diel vertical migrations, during daylight hours, most species remain within the bathypelagic zone between 300 and 1,500 meters deep. Towards sundown, fish begin to rise into the epipelagic zone between 10 and 100 meters deep. Lanternfish do this to avoid predation, and because they follow diel vertical migrations of zooplankton upon which they feed. After a night spent feeding in surface layers, lanternfish descend back into lightless depths gone by daybreak. By releasing fecal pellets at depth, lanternfish make the carbon capture process called biological pump more efficient. Due to gas bladders, these layers are visible on sonar scans, giving the impression of a false ocean bottom. This is the deep, scattering layer that perplexed early oceanographers. Great variability in migration patterns occurs within the family. Some deeper living species may not migrate at all, while others do so only sporadically. Migration patterns depend on life stage, sex, latitude, and season. Research published November 13, 2024 in PLOS 1 provided new morphological analyses of complex structure of primary photophores from representative species in 17 genera where photophore morphology had not been previously described. Histological techniques revealed considerable variation among species in specific aspects of photophore morphology. Secondary reflectors were observed in several species including Benthosima suborbitale, Lewena rara, and Symbolophorus evermanni. All 4 species with secondary reflectors also possessed associated secondary pigment layers. Researchers hypothesized the presence of a secondary reflective layer. An associated secondary pigment layer affects both direction and intensity of emitted luminescence. Since bioluminescent light is reflected off the layer of guanine prior to emission, characteristics of the reflector have a priority effect on spectral quality of light emitted. 4 additional species contain 2 visual pigments. Mathematical modeling based on assumptions of sensitivity hypothesis showed that, contrary to belief that deep sea fish's visual pigments are shortwave, shifted to maximize sensitivity to downwelling sunlight, visual pigments of myctophids instead seem better placed for visualization of bioluminescent signals. Research published in Philosophical Transactions of the Royal Society B showed lanternfishes possess optical sensitivity to extended light sources 10 to 100 times higher than the human eye. Estimated global biomass of 600 million tons makes them one of the most abundant vertebrate groups on Earth. Their position in the water column and vertical migrations mean myctophids play a prominent role in ecology and energy transfer within the mesopelagic zone. They account for over 60% of all deep sea fish biomass. Despite ecological significance and large biomass, remarkably little is known about basic biology and ecology of lanternfish. During April, spring, and July summer surveys, they found these fish consumed primarily copepods during morning and evening hours, with those in the genus Oncae comprising as much as a third of all items. They occasionally consumed amphipods, mysids, which are opossum shrimp, and ostracods in small proportions, less than 3%. In the epipelagic layer, skinny-cheek lanternfish do not typically eat more food than 1/3 of their body weight. In search of zooplankton, lanternfish are able to hide from predators while migrating vertically. They camouflage themselves and appear as a mere shadow. In the Arabian Sea, predators such as Alcock's bowfish Stomias nebulosus, Sloane's viperfish Chaliotis sloanei, and viperfish lacking a common name Chaliotis pamellas are known to prey on myctophids primarily. Additional predators include penguins, non-mesopelagic fish, and other lanternfish in family Myctophidae. Others, through specialized light organs in their bodies called photophores, some do both. Reasons are as diverse as methods: camouflage, defense, communication with fellow shrimp. Research led by Stormy Collins, a PhD student at Florida International University, and Associate Professor Heather Bracken Grissom, concluded bioluminescence is much more common than previously thought, not only within shrimps, but across all deep sea animals. Research published March 5, 2025, showed these shrimp evolved visual systems with a diversity of special light, detecting proteins called opsins that help navigate their bioluminescent world. The proteins allow shrimp to see a range of colors, including environmental and bioluminescent blue light. They might even differentiate between their own glow and bioluminescence of others. Shrimp that migrate into shallower waters, where there is more available light, have greater variety of light-detecting proteins. This is likely because they need to see a wider range of colors to navigate and survive in more complex light environment. Research presented at the 17th Deep Sea Biology Symposium examined visual evolution of deep sea shrimp. Bracken Grissom is a marine evolutionary biologist who was the first to identify an unknown species of monster larva, part of an international team that captured the first ever video of giant squid in U.S. waters, and has both a species of crab and shrimp named after her. Research from 2024 on crustaceans, collected with trawls from deep ocean water, column off Southern California, continues to study biodiversity. Factors affecting distribution of bioluminescent bacteria include temperature, salinity, nutrient concentration, pH level, and solar radiation. Bioluminescent bacteria are found globally as free-living symbiotic or parasitic forms, and possibly as opportunistic pathogens. The catalog published March 24, 2024 by researchers at Université Catholique de Louvain in Belgium represents the most comprehensive and informative listing of luminescent marine animals ever published, based on latest taxonomic information and associated with an innovative luminescence scoring scale. It shows world's luminescent organisms now encompass approximately 1,000 genera, a significant increase compared to previous estimates. It consists of a robust foundational tool for studies in or related to the field of bioluminescence. More than 350,000 observations classified for bioluminescence capability based on literature descriptions. 553 phylogenetic concepts distributed within 13 broader taxonomic categories. While some estimates of proportion of bioluminescent organisms in open ocean were previously published, that study was the first based on thoroughly quantified data set for full range of midwater taxa. The most complete list of bioluminescent taxa in literature was published 30 years before that study, with few additions since. Because there is fairly low level of activity in deep sea research, and even less work on potentially bioluminescent organisms in good condition, rate of discoveries of bioluminescent taxa occurs at a very slow decadal rate. Because it occurs across diverse taxa, measurements of bioluminescence can be powerful to detect and quantify organisms in the ocean. 3/4 of animals observed produce their own light. Years ago, during the Cambrian explosion, when eyes first evolved, eyes and bioluminescence co-evolved. Vision did not come first to see the sun, vision came to see each other's light in the dark. The ocean invented its own stars before it had a reason to look up, and these 7 creatures are not just survivors. They are the architects of an illuminated world that most humans will never witness. That is the reveal. The ocean does not borrow light, it makes it. And it has been doing so since before there was anything alive to see.